Now there are three common types of restriction modification system. There is fourth type, also called type 2s, but most of the textbooks deal about these three types of restriction endonucleases, among which type 2 is the restriction endonuclease that is used for genetic engineering because it is highly specific. It exactly cuts at the site for which it is assigned for. So because of the character that it has a separate endonuclease and methylase activity, but type 1 and type 3 they are multifunctional. Type 1 is a single multifunctional enzyme, and type 3 is in fact there are two subunits which are associated together by another subunit of protein, but the whole enzyme is multifunctional, that it has the restriction as well as the modification or methylase activity together. But type 2, these two activities, endonuclease and methylase activities, are separate enzymes, they are not same. So while type 1 and type 3 they try to bind with the restriction site where they have to make a cut, what they do, they modify the same site of the DNA with the methylase activity, and this covalently modified site is not suitable for the binding of the endonuclease part of the enzyme, and it slips slips aside, and it introduces cuts away from the actual restriction site. In case of type 1, it introduces cuts 1000 base pairs away from the actual site, whereas the type 3 it introduces cuts on the 3' side about 24 to 26 base pairs away from the actual site. So type 1 requires only magnesium, it does not require S-adenosylmethionine and ATP. S-adenosylmethionine is the substrate for is the methyl donor actually, which is required by the type 1 and type 3 restriction and modification systems, and because they actually methylate the site where the restriction has to be made, and they also require ATP for that. So these are the property, and the last one, that this type 3 is it introduces symmetrical cut, or it digests the DNA in a symmetrical, it is a dyad symmetry. It splits the DNA into two parts where either it produces sticky ends or blunt ended DNA, that we will see later on. But the other two, these enzymes type 1 and type 3 restriction modification system, they have an asymmetric ends that they produce after digestion, and they produce actually digest the DNA at non-specific region, so that those cannot be used for cloning purpose. Type 4 or the type 2s is a type of restriction endonuclease that can cleave methylated DNA. That is another interesting thing, that can cleave methylated DNA, and so it can probably be used for understanding the epigenetic part of this molecular system. And there are some examples like the MCRA, MCRBC, MRR, isolated from Escherichia coli itself, are the examples of this type 2s restriction modification system. Among these four types of restriction endonucleases or modification system, type 2 is useful for the purpose of cloning or genetic engineering, you can say. Nomenclature of restriction endonucleases. A suitable system of nomenclature of restriction and modification system was proposed by Smith and Nathans in 1973, and a simplified version of which is used nowadays. Restriction endonucleases are designed by a three-letter species identifier written in italics, followed by a strain identifier and a Roman numeral. For example, the restriction endonuclease named EcoR1 is derived from the first letter of the genus Escherichia. E is derived from that, the first letter of Escherichia, and the later two letters that the CO is derived from the species coli. The letter R is derived from the strain RY13, and the Roman numeral 1 is is because this enzyme was probably the first to be discovered in the system. But sometimes these Roman numbers they signify the source from where this enzyme was obtained, was actually acquired by the cell, whether it is a plasmid generated or path generated. On the basis of that also these Roman letters are being used sometime in the name of restriction endonucleases. So similarly, Hindi 3 is it indicates that it is being present in Haemophilus influenzae strain D, and it was the third enzyme isolated from that bacterium. There are homing endonucleases that are named in a similar fashion, except that intron encoded endonucleases are given the prefix I, for example ICU1, and intane endonucleases have the prefix PI, PIPSI. Where it is necessary to distinguish, whether it is necessary to distinguish between restriction and modification system, they are given the prefix R and M respectively, as for example R SMA1, M SMA1. So these are the way how restriction endonucleases are being named. And now talking about the properties of type 2 restriction sites, or this is about sites where the restriction digestion will take place. Type 2 restriction sites generate symmetry. However, if the cleavage positions are not directly opposite to each other, like this one, this one, here the cleavage positions are not opposite to each other. So in that case, they will generate sticky ended or staggered cuts like this. You can see the TTAA, this portion is the overhang or the sticky part of the cleaved DNA, because the covalent, because the phosphodiester bond that has been cleaved is not exactly opposite, it is 4 nucleotide apart from each other. But here in this case the SMA1 enzyme, it clips the DNA at this position between this CCC and GGG on both the strands, and the cleavage sites are opposite to each other. That's why it produces a blunt ended DNA like this. But blunt ended DNA are not sticky, so you need to modify them so that they become sticky. We will see how linkers are being added or ligated to these blunt ended DNAs to make them sticky. Majority of type 2 REases, means restriction endonucleases, recognize a particular sequence in the DNA where they bind and make specific cleavage. These sequences are popularly known as the palindromic sequence or the palindromes, because they read same from the both ends, like the madam, I am Adam. The restriction sites, however, read same by their complementary ends, like this one, for example the GAATTC. If you go by this one, the pi prime GAATTC 3 prime, the opposite strand is also pi prime GAATTC 3 prime. Similarly, this one CC pi prime CCC GGG 3 prime, the other strand is again pi prime CCC GGG 3 prime. So all restriction endonucleases have this kind of palindromic sites where they will bind and they can make cleavage either of this. They will either produce a staggered cut or will produce a blunt ended or plush ended DNA.